Hello, welcome back to part 2 of our look at action potentials and exactly how we initiate an action potential. Now that we understand what an action potential is, let's actually look at what initiates the action potential. And so to start that, we have to understand some terminology. And so the first two pieces of information that we need to understand is this idea of a ligand and a receptor. This is very similar to what we looked at when we talked about a substrate and an enzyme. So if you think of the enzyme as being a receptor, something that is going to receive, and if you think of the ligand as being the substrate, the thing that is going to bind to, this will be a pretty easy concept for you to understand. The ligand is what will bind to the receptor, and for us, the ligand is a neurotransmitter, because the neurotransmitter will bind to a specific receptor. All right, and the neurotransmitter, generally referred to as a ligand, has to have specificity. Right? In other words, specific ligands can only bind to a specific receptor. And when that does, it's going to cause a change in the shape of the protein or the receptor. All right? And by binding to the receptor and causing a change in the receptor, you open the sodium channel. Right? That sodium gate then opens up. And we've looked at this. I've made sketches on this back on the previous videos for muscle physiology, and we will revisit that just to make sure that you're seeing where this is happening. But where I am referring to specifically is within the junctional folds, where the axon terminals are connecting or communicating with the sodium channels inside of the junctional folds. All right, so maybe that'll ring a bell as far as where I am referring to within this whole process. And as I just mentioned, ligands are, or a type of ligand is a neurotransmitter. All right, and the receptor is what we define as being a neurotransmitter receptor. All right, and as we've already looked at and talked about, we know that acetylcholine is the neurotransmitter that will initiate muscular contraction within skeletal muscle. Acetylcholine is the neurotransmitter that is going to initiate the action potential sequence that's going to travel down the sarcolemma, down into the T-tubule, that's going to stimulate the opening of the voltage-gated calcium channel, that's going to allow calcium to leave the terminal cisterns, go into the sarcoplasm, bind to the troponin, spin tropomyosin, uncovering the myosin binding sites on actin, allowing actin to bind to myosin if ATP molecule is bound to myosin, and the story continues. Right? This is where that acetylcholine is initially coming into play to create that first action potential that is going to travel from sodium channel to sodium channel down the sarcolemma. Now, that acetylcholine is going to open a chemically gated sodium channel. So the sodium channels inside of the junctional folds are chemically gated. The sodium channels along the sarcolemma are voltage gated. What's the difference? The difference is what stimulates them opening. In the junctional fold, acetylcholine is a neurotransmitter. A neurotransmitter is a chemical, and so therefore those sodium channels are chemically gated. What allows them to open is the binding of a chemical. The action potential is generated. The action potential is an electrical charge. It's an electrical current that is now traveling down the sarcolemma, and it comes to a sodium channel in the sarcolemma that is opened. That is an electrically gated sodium channel, because what stimulates the opening of that sodium channel is an action potential. So the difference between a chemically gated and a voltage gated sodium channel is A, location. Chemical gated are in the junctional folds. Electrical gated or voltage gated are along the sarcolemma. And what precipitates the opening of the sodium channel. The chemical gated sodium channels in the junctional folds are opened by a neurotransmitter. It's a chemical. The voltage gated sodium channels along the sarcolemma are opened due to an action potential, which are voltage. It's voltage. It's electrical energy. All right. So that, I think, is making sense, all right, to everybody, hopefully. And of course, the sodium and the potassium voltage-gated channels, they are going to open, or when they send the action potential, that is what we define as being threshold, which we already know is negative 55 millivolts. Right? And we know that that action potential is an all or none concept. It's an all or none premise. Right? The action potential is either sent or the action potential is not sent. And if it is sent, it's always sent. The strength of an action potential is always the strength of an action potential. All right. And then again, what's left behind after the action potential leaves, after the neighborhood kids leave, is the repolarization and the hyperrepolarization, returning back to resting membrane potential, and that's what we see here in this diagram. All right. So inside of the junctional fold, all right, there is a what we call a synaptic cleft, or a synaptic junction. In other words, there's a space that allows the neuron, the axon terminals, to communicate with the channels that are found within the junctional folds. And to understand that, this is what we need to look at next. When a neuron communicates with a muscle cell, that space, that gap between the axon terminal and the junctional fold is what we call a neuro-effector junction. All right. That is a little bit different. It's the same premise, but it's a little bit different than what we refer to between the communication between a neuron and a neuron. So if a axon terminal was to communicate with a dendrite, we would call that space a synapse. Right? Just like what we've talked about in lab. When that same axon terminal is communicating with a junctional fold in the sarcolemma, we refer to it as a neuro-effector junction. And so, what is happening inside of our neuro-effector junction? Well, this is a 5-step process. Right? This is a 5-step process. And so step 1, an action potential is going to travel down the axon of the neuron into the axon terminal and into the synaptic end bulb. Right? Into the synaptic end bulb. We're going to talk about how exactly an action potential travels down an axon in the next video when we get into the nervous system a little bit more. But for right now, know that that action potential is also traveling down the axon, reaches the axon terminal and the synaptic end bulb, and inside of that synaptic end bulb there are what we refer to as vesicles, and those vesicles contain the neurotransmitter, in this case acetylcholine. Step 2. There are voltage-gated calcium channels inside of the synaptic end bulb. Right, so inside of the synaptic end bulb, there are voltage gated calcium channels. So I'm going to kind of draw this in here. Right, I'm going to draw this a little exaggerated. So this is the axon. This is the end bulb in the axon terminal. And inside of here, and so our action potential is traveling down. Let's do that first. Action potential traveling down. Here's our action potential. It reaches here, and it causes calcium to be released. Causes calcium to be released. And so here is my calcium that is being released. All right. So if I blow this up a little bit bigger, if I take this and blow it up a little bit bigger, inside of our synaptic end bulb, calcium is being released. Right. And that calcium is causing the vesicles to bind to the plasma lemma to release the neurotransmitter into the synaptic cleft. So let's come back into here. Right, so now we've got these vesicles that are now being given permission to bind to the plasma lemma. So they bind right here. And when they do so, they release acetylcholine into the synaptic cleft. Right? So it crosses over the space. And in step 4, that neurotransmitter, again, remember that the neurotransmitter that we are referring to is acetylcholine. All right, and so acetylcholine is going to cross that synaptic cleft. It's been released from the vesicles, which were stimulated to bind to the plasma lemma by the release of calcium that was stimulated by an action potential that was traveling down the axon and reaches that terminal end bulb. And in the final step in this process, the action potential is stimulated because the acetylcholine binds to the sodium receptors in the junctional folds. And that sends the action potential down the plasma lemma, going from sodium channel to sodium channel. Every time an action potential reaches a sodium channel, the new action potential is generated. All right? So know that it's not the same action potential that is being created. It is a different action potential that is being created. It is a new action potential that is being created. And I think on the student slides, I think I have an animation and a video for you to watch on this, just to kind of give you a little bit more clarity. But the acetylcholine stays bound to the sodium channel in the junctional fold until it's removed. Right. Acetylcholine stays bound to sodium channel receptors in the junctional fold until it is broken down. All right? What breaks down acetylcholine is an enzyme. And the enzyme that breaks down acetylcholine is called acetylcholinesterase. Acetylcholinesterase is the enzyme that breaks down acetylcholine and causes it to not be bound to the sodium channels in the junctional folds anymore. Why is that important? Because if acetylcholine is not bound to sodium channels in the junctional folds, a new series of action potentials will not be created, which means you will not have a muscle contraction. All right, so acetylcholinesterase is critical in breaking down acetylcholine to prevent an unwanted action potential that will eventually lead to an unwanted muscle contraction. And that is the whole story. That is the beginning of the story on how our muscles contract. And so as you are reviewing and studying and digesting all of this and asking questions, I wish you continued success, and I will see you on the flip side.